Hi, my name is Mudita Vats. I'm a PhD student at M4I Maastricht University, the Netherlands, with Ron Hieron and Berta Sierra Pesto. Why and how I came from India to the Netherlands is a story I'm gonna tell them. When I dreamed about becoming a scientist, I was doing my bachelor's in microbiology, and I felt I should not do research to get appreciation from scientific community or to have huge list of publications which a person from different background cannot understand, or in general society gets no benefit out of my research. So I kept exploring different areas to know the answer to my question, that what will be my focus. And I participated in different workshops, trainings, and all I could do. And I came to the conclusion that I want to do something in environment or food safety, or maybe both. But then this went in the background, and I went on to do MSc in applied microbiology. Then I got some experience as a junior research fellow in a pulp and paper based research institute. So before I move on, I want to tell that I am an early career network lead on MSACL. I love to travel, dance, and play basketball. My past experiences form a foundation of my PhD. And so during my master's, I went to CSIR CCMB, Hyderabad, India, and I was studying the formation of B1 ions in chemically modified peptides over unmodified peptides. We could validate 673 proteins out of 1070 proteins, and this work got published in European Journal of Mass Spectrometry. This way, I got some insights on mass spectrometry, and I decided that I want to do a PhD which includes mass spectrometry. Then my mass spectrometry story took a break, and microbiology said, hey, don't forget me. I was working as a junior research fellow in Central Pulp and Paper Research Institute, where I developed methods to determine microbial load in food packaging paper and paperboard. We could identify some novel microorganisms when we were looking for the predominant microorganisms by using 16S rRNA sequencing. Besides this, we were doing other projects and tests. For instance, some manufacturers that claim their paper to be antimicrobial, we took their paper and tested for antibacterial and antifungal properties. We were trying to produce bioethanol using lignocellulosic waste, which is an environment-friendly alternative to chemically produced ethanol. And this also reduced the burden of waste on Earth. And then I was part of a project where cellulose was produced using bacteria. So it was bacterial cellulose, which, if incorporated with paper during the manufacturing of paper, can give tensile strength to the paper. What was going on in the background of my FoodTraNet application? I was impressed by the research infrastructure of M4I, and when M4I is collaborating with FoodTraNet, I was not the one who was planning to miss this opportunity. And then if I talk about how my past brings me close to my present and future, so my experiences in microbiology and my enthusiasm to do something to food safety and environment and bring all of them close to mass spec and mass spec imaging brings me here. And another happy thing about this PhD is the international environment provided by FoodTraNet and M4I both. We are people from different countries, and I really enjoy that. And then I want to come up with environmental friendly alternatives to control pest, and I'm here to broaden my horizon. Apart from getting a degree or bringing a change in current scenarios, I always make sure that I'm happy with what I do. If I am not, then I just leave. So here I will be developing new methods to study the influence of organic versus chemical fertilizers and to look at plant microbiota, because these might be novel organisms which will make the pest prevention environment friendly and a good alternative as compared to the chemical fertilizers. The technique that I will be using include but is not limited to Marley, Desi, Rhymes, LCESIMS, and I'm really curious to see how efficient Marley biotyper will be, and will we be able to replace it with 16S rRNA sequencing to reduce time and effort. So right now, I'm getting trained on some of the mass spec imaging techniques, and this picture shows a 3D scan of leaf. This was performed by Sylvia, who is another PhD student at M4I, and I'm slowly getting trained on this to become independent. This picture shows the 3D scan of mushroom, and I am taking this, apart from my main PhD project, I'm taking mushroom as a model to optimize the sample preparation strategies for mass spec imaging using Mudita Vats and Rhymes. My long-term goal is to see myself as a scientist in industry, and I want to bring microorganisms close to mass spec imaging, and then I want to make food safer. I thank FoodTraNet and M4I, and I will together work with them to bring myself close to my dream. And your questions are welcomed. Thank you.